Hey, Avahan. Hello. Avahan, can you hear me?
Yes, I can hear you.
Okay, excellent. How are you doing? How are you getting on? I tried to comment on your introduction as much as I could, but it looks really good. It started getting into shape. Do you have any questions in the meantime for your feedback?
Yeah, I have one.
Excellent.
So I wanted to ask, like, if we have like two sentences from the same paper, then like, should we reference like each sentence with the like same name, like same reference, or can we like combine and do like one reference at the end of the two sentences?
So the general rule is you reference everything you say, and normally you would try to, use as many references to, not have the repetitive it's okay to use a reference, let's say 2-3 times in your like introduction or your discussion, but more than that you try to find another reference. That's like a general rule when you write a paper or thesis. But I think in your case, because you've got like limited number of references, you can reference the single reference multiple times. It's fine.
Okay,
Does
so
that
if
make
we
sense?
have like two continuous like lines that are from the same paper, so we should write the same reference in both, right?
It
Like in
depends
each
if you're
sentence.
making a separate point, Avahan. So if each sentence has a different point, then yes. If like the
Okay.
first sentence is continuation of the other, so you kind of build on what you said in the previous sentence, like, then it should be fine to just reference the last sentence. Does that make sense?
Yeah, I got it. Thank you.
Yeah, okay, cool. Yeah, I know I added some referencing. It's just normally, if you write anything in science, you reference everything you say, unless it's like a given knowledge, let's say. And at your stage, writing that, you probably nothing is given knowledge. It's all quite new for you, right? So you're learning about it from scratch. Let's say if that would be an article, there's sometimes, everyone in the field know it. So you don't have to necessarily reference that sentence. But overall, you reference everything you say.
Yeah, I got it. Yeah, thank you.
Yeah, excellent. Hey, Hailey, how are you doing?
Good, how are you?
Yeah, I'm not too bad. Thank you so much. I saw you send me the screenshots of your intro. I'm not able to read that properly. It's very poor quality. If you could share a Google Drive with me, that would be very useful. So rather than, because you wrote it.
I can share the document with you right now.
That's great. Yeah, and just allow me for editing, and you can send me the link on Slack so I can, after the session, sit down and have a look through it.
Okay, I think it should just go to your email. So I think it'll just share into your email and you'll see it there.
Okay. Let me just because you can click share and then you can copy a link. So if you send me the link, that will be easier, because then I request the privileges and then you can allow me to do it. That's how we done it with Avahan as well, I think.
Okay, yeah, I think I can just give you them though, like through Google Docs. I don't know if you've ever done it that way, but it just shows up and you should be able to edit.
So I need to give you my Gmail then in that case, I think. I'll send it to you now. @gmail.com. Okay, I send you my Gmail address so you can just do that. Share it through there if that's okay. Let me know how you get on. I see if I get the email.
Where'd you send the email?
On Slack?
Okay, thank you.
No worries. I just send it in direct message to you. Because if you share it with my Cambridge University email, I'm gonna have to request access again, because it's not going to link automatically. Cool. So Hailey, you managed to write your intro from what I see, and you started on the results as well, right?
Yeah.
Okay, cool. And so there's 3 parts of the results. You managed to write up the first part.
I skipped the first and did the second, but then
Okay.
I'm gonna go back and do those.
Excellent. If you can get the first one done by the end of the week, then I could look at all together and give you feedback by Monday. How does that sound?
Yeah, I was gonna try to do like all of the results this week. Then I have a question, like what are we supposed to do? We're supposed to like make charts. I
Yes.
wasn't doing that and I don't really understand that.
So yeah, we can go through that. Like I think I've handed some tables. I can't remember if you added some plots. Can't remember now. Let me have a look at it.
Like tables of what am I supposed to be?
So you're trying to show your data in the most like kind of efficient way, so someone can easily look at it and be like, okay, that makes sense, right? So have a little think. So for the first part, you're comparing different formats from different websites, right? So how would you think would be the easiest to compare that, to show to someone the differences? Obviously, you describe it in text, but maybe like a table with all the different components of the files would be useful. What are the differences? What are the similarities? Okay.
Yeah, that makes sense.
That makes sense,
So
right?
do we have to do that for all 3 results or just like one of them or just like 2?
I would try to do the tables for all your results. You can, because I think tables are useful. So if you're comparing different sequences, then you can, let's say, put the excision number in one column, then the strain name, then the size, the length of your gene, and all those kind of information there. So, and then you can discuss it in text. So for the first part, I wouldn't probably do a figure, because most of those sequences, we expect them to be similar. So there won't be that much information to, kind of analyze. You're just doing straight comparison. But the second part, when you're actually looking at the sequences from different, let's say, different Staphylococcus aureus or different species, then it's nice to have that included in a table.
So do it for all 3 results?
Yeah.
Okay.
Yeah, so this generally, like Ash said, I don't know if you guys saw, and Ash sent like a nice on general chat on Slack, I think, or questions. He sent like a summary of all the results on Slack. I don't know if you saw that. On general chat. So if you go to general chat now, you've got Ash summarize for you what you need to do for the report. Just again, the structure and the different results. I know he doesn't explain how to do figures and tables. We can go through that quickly again. We went through it last week a little bit. But generally, like, you try to make the figures to kind of show the data in a more logical way than you write in text. Because sometimes, we look all for patterns as scientists, right? We look at something, we look for patterns, and we try to understand what's going on within the results. And sometimes having a figure really helps to get your head around what's actually trying to say. So it's just to make it as easy as possible. You can always describe it in text, every complicated result. However, sometimes it's just very hard. I've got duplication. Avahana, you here twice. That's interesting. Cool. So yeah, so basically it's just the figures and the tables just help you a little bit better to understand what's going on. Does that make sense, Hayley?
Yes, that does. Thank you.
Yeah, so yeah, just give it a go, have a little thing. Once you have it done and written up, and I have a look. So I try to look at over the weekend. So if you can write some of the results, maybe not necessarily the third part, but at least the first and second. So I give you some feedback in the Google, and then we can, if there is any questions more on that, or you're confused about how to do it, you can always discuss it. We've got still 2 sessions left. So the next session would be more, looking at the result. That's why I'm trying to get you to send it to me before the next session, because the next session already will be Saturday, if I'm correct. So we're not meeting on Wednesdays anymore. So there's 2 more sessions on Saturdays, I think, according to the plan. That's what Ash said as well. So there will be 2 more sessions. So I think the 1 to last session would be nice to have the results and introduction all completed, because the discussion will take you a little bit. Discussion is very hard to write. Discussion, I personally find the hardest to write. Results are easy because you do analysis, you write them up as they are. Introduction is fairly easy because you know what you want to talk about. Discussion, you try to put your results in the context of the literature. Right, so wherever you found, was that found before? Is that surprising? Or is that kind of confirming what literature suggests? So try to discuss those findings in the context. Obviously, with like different browsers, the first part, there would be that much to talk about, but the part 2 and 3, you're going to have results of different comparative work between genes. So you can, try to understand the diversity using your results and try to look in the literature what the diversity is expected. So if you see, I don't know, nucleotide mutation, one nucleotide substitution, and you can look into it and see if that has a function, if there's a functional change, if there's amino acid change. Let's say if someone in the literature already discussed that kind of amino acid change, and does it have an effect on the functionality of the gene? Okay, does that make sense?
Yes, I had one question. Like, should we limit our like figure and table like one per one result, or we can do more?
Not necessarily. No, I don't think there's a limit to it. Obviously it has to be kind of proportional. So if it's all tables, it looks a little bit weird. But I think there's this thing you do as well in papers and writing thesis. Occasionally you have main tables in your text, and then you get additional tables which are not key, but you wanted to add the data for someone else to look if they want to. And those are called supplementary figures and tables. Have you heard of those? You came across it in a publication when they said supplementary figure or supplementary table 1 or 2?
Yeah.
Yeah, you've seen that. Yeah, so those are basically those which are not super key, because often journal gives you like limitation. They just want 3 tables and 5 figures. So if you have more stuff, then it has to go to supplement. So you only select 5 main things, main results, and then everything goes into supplement. I don't think we're going to do that. But if you feel a table or figure is a little bit out of space, but you still want to include it, then you can always add it at the end as a supplementary figure or supplementary table. Okay, so you don't have to worry about doing too much or too many tables. Sorry, Rana just messaged. Let me just hand her a link again. Give me 2 seconds. I think the Zoom is being a bit tricky recently. Okay, Rana should join. So yeah, so you can add this additional information into the supplement. I think that's fine. And then the way you reference to those tables or figures in a text, I tend to call it table S1 or figure S1 as a supplement. But you still need a proper figure legend for those supplementary figures as well. And let's say table legend. So I don't know if you, Hayley, were here last week when we spoke about it. I don't think you were. But basically for figure and tables, for figures, the legend go on top of a figure. So you call it a figure 1. Then in the first sentence, you describe the main finding in your figure. And it doesn't always apply because sometimes the figure doesn't show like a key result. If it's a key result and there's a key figure for understanding your results, then you want to like the first part of the sentence would explain what the figure is showing. So the main result. And then after that, you explain how you designed the figure, what the figure contains. If there's any shortcuts or if there's any colors or things you need to describe, then you do it in a figure legend. Does that make sense?
Yeah, do you think you could like get an example?
Sure. Let me show you my figures. They're not published yet, but I can show you some of the figures with the legend. Give me 2 seconds. Let me just bring it up. Let me show you a review. 2 seconds. Okay, so that's not published yet. So just letting you know it might change on the way and stuff. So, okay, here's the figure. Now, again, you're not going to have that kind of size of figures and that big. Can you see that?
Yes.
Okay, excellent. So we've got phylogenetic tree. So that's the so you can see we call it A. So that's the figure A, and then we've got B, C, and D. Okay, so it's just easier to refer to it to text. So you kind of, if you have multiple figures, then you do them A, B, C, D. It can be capital, it can be smaller case. Then you've got the figure, then you include figure legend here. So you'd explain what each colors correspond to. Same here, you know, you've got the axes. So you've got all the axes and you explain what those are. Here's kind of obvious because that's a year. So you don't have to put a year here. Here's kind of obvious as well because it's host and country. And then on the bottom is count. Does that make sense? So those are the axes. You've got heading for each of the figures if you've got multiple, and you can see here's the legend. So this is the S1. So it's like a supplementary figure. So you say the temporal host species and geographic distribution of our collection of CC398 isolates. Yeah. So it covers, so we've got the countries, we've got the host, and we've got the dates, right? Those are the things. So temporal is basically a date. Then we've got A, so small a, and we explain it. So it goes from small a, so that's the small a. Then we explain phylogeny of CC398 with rings showing the host species and countries of origin for each isolate, number of groups, you know, smaller, larger than 10 isolates shown. So anything smaller than that per country was excluded. And then we say the blue outline indicates the LA associated. So we can see it here, right? The blue outline we explain. And the red outline indicates the 5 most recent isolates in our collection. So I've got the red ones here. You can see that. Sample 2018 from pigs on UK farms. Then we've got B, C, and D show the variation in sampling date, host species and country across the livestock associated and human associated clades. Does that make sense?
Yes, thank you so much.
Yeah. Yeah, so it kind of describes the figure, what's happening. Obviously, you don't go into through the details because you've got a legend here, so you don't have to say, oh, you know, the isolates were sampled from those hosts, because that's kind of, you can read that from the figure. But it's just someone, if it looks at it, it knows what each of the aspects of the figure means. Okay, and same with tables. You would have, I don't know, I don't have tables here, but it would be same with a table. Let's say in a table, you have to think about it. You know, if someone doesn't know anything about the project, anything about what you did, can they look at the table and understand the values? Okay, so like a p-value is a standard way, so you don't have to explain that. But let's say you named your column COV underscore, and that's coverage. Okay, so you named it like a short way. You have to explain it in your figure legend what that refers to and how did you calculate it. Okay, so you're going to do like that. You're going to do the direct comparison between sequences. Hey, Rana. You're going to do the direct comparison between 2 sequences, and then you're going to have different parameters, you know, which describe how similar or how different those 2 are to make a gene. So you're going to have the length, you're going to have the number of nucleotides, you know, what amino acids are they translated to, and so on. So you're going to have a range of different things about the 2. So table would be nice to compare it, right? Cool. Yeah. Great. Yeah, so give it a go. And again, I can look through it and make sure it's all there. But having some simple tables and figures, I think is a good start. And again, you saw my legends was quite long. So explain it because it was a complex figure. If you have a simple figure, sometimes 2 sentences is enough. One which describes what the table is representing. Second one, what each of the parameters of the figure or columns in the tables refers to. Okay, cool. Rana, do you have any questions?
Yeah, I had a question about the first part of the results. So like I wrote the yeah, are we comparing like the databases and like their qualities and like their features in the first part? Because like I did comparisons in between the strains of like S. aureus for my first part.
Yeah, that's the later part, right? That's later you use the actual FASTA sequences and you compare them. So you did that. So you did different Staphylococcus species or different Staph aureus strain, so that's great, you know, so you had like an idea what to do there. But I think the first part, and I know it's not the most exciting part. Avahan did it very well. Avahan, do you mind sharing your screen with your comparison between the different files, because you did it very well. I think it's nice for other people to maybe see, if you don't mind.
Yeah, I'll do that.
Great, thank you so much. So yeah, he wrote it up it's not exciting data, like I know it's just file formats, but since it kind of helps for you to understand what are the differences and how to use those and apply. So it's not going to be the most exciting section, I'm aware of that.
All right, and
But
like I have another.
that's a minimum. So there's minimum requirement. So I don't know if you saw, Rana, on the actual Slack channel, in the general chat, Ash put yesterday, I think, like a nice summary of what you have to do for the project. So just read through that. Okay.
I was just like going to ask, yeah, like that's a really great idea to do, like the
Table.
graph.
Right?
Yeah.
So that's what Avahan did. So he did a table with those symbols, which things are present, which things are identical, where are they places, like a nice little comparison. I wouldn't make it better. So he done it in a table. I think this is very, very good. As I said, you can use like presence absence instead of the symbols, but the symbols are quite nice to see, right? Because they're quite obvious. You probably wouldn't do that in a scientific paper.
All right.
But you would probably do, you know, plain text rather than symbols, but it's absolutely fine for the purpose of that.
All right. And I had another question. So in my results part, I decided like maybe it'll be great because like I'm gonna focus more on the prevalence of the MECA gene. Maybe it'll be better to like first
Thank you,
look
Avahan.
at
Thank you so much.
thanks, Awahan. Well, I thought maybe it'll be good to first look at the S. aureus strains and then the prevalence in other species using BLAST, and then using like the UniProt. And I also used BLAST for like the protein sequence. So, and then I found like a bunch of other species that had hypothetical proteins, like not specifically the penicillin binding protein, but like hypothetical proteins that were similar to the PBP2A. So maybe like it would be good to kind of like.
No, that's a really cool idea. Like, just make sure you've got word limit. Okay, so do all the basic analysis first. So you've got set of basic stuff Ash asked you to do. So do that first, you know, write it up. It can be short and, you know, as long as it's there, it's fine. Then you can build on that. If you have space, if you have the words left, I think then you can make it a little bit your own. And also the point 2 and 3, is a little bit more freedom there, right? Ash doesn't specify how many sequences you have to look at, what parameters you have to look up. You get the freedom to do that. So I think from part 2 and 3, you can like take it own and do comparisons. So you can have a result section, you know, comparative.
All right.
You know, comparison or diversity within the MECA gene in Staphylococcus aureus, and then you can say I selected 20 or 10 sequences, you know, for those strains. So you can do a table there, so you can have excision number, strain, species, gene length, you know, all this stuff. So that would be table of all your sequences you selected to analyze. So that would be your first table, and then you can look at the differences in alignment software as you learn to do it, or you can look at the amino acid sequences, or you can even look at the protein. So then you can do another table, like more likely comparing them directly.
All right, that sounds great. Thank you.
Yeah, if you identify a SNP in, let's say, and there's that's the nice thing to do as well, I think. If you identify a SNP and it results in amino acid changes, that's worth describing in. Avahan did that as well. So, you know, having a nucleotide change if it's synonymous or non-synonymous mutation, okay? And then you can describe that. So you can suggest potentially this could have a impact on the protein structure if it's amino acid change. And you can even, on UniProt, you can even look at the different domains of the protein, right? So you've got the protein, you load it, and then there are functional domains you can look at. I don't know if I showed you how to do that.
Yeah, like we did at the last session.
Yeah, okay, great. So yeah, so you can just do that and say, okay, this amino acid change occurs in that important functional domain. So it could potentially result in some kind of change in functionality. If it's outside of the domain, if it's like at the end, what happens if it's at the end of the gene? What would you think it happens if it's at the end of the gene?
Probably like if it's at the end of the gene, it wouldn't really impact unless there's an amino acid change.
Yeah. Even if it's amino acid change towards the end of the gene, it tends to, the way the genes usually are, the first part of the gene is very important, the middle bit at the end is usually, if there is deletion towards the end, or if there is mutation, doesn't tend to change the protein structure as much or as such. Obviously there are exceptions, but it tend to not be associated with huge change then. So there's something to think about, and then describe the mutation, potential differences. And you can do the same if you do between species and you're interested in that. You can, I don't know, do 10 per, select, let's say 2 or 3 species, which you know have MEC-A, and then look at the differences between them, right? So do they have to tend to have the same SNPs? So if, yeah, I don't know, stuff like epidermis, I think has SCMEC too. So if epidermis has, you know, associated the same SNP, which is different to all the other Staph aureus, you know, that, and then you can discuss it. Most likely this mutation, you know, either appeared just before the SCMEC entered the species or within that species, right? It didn't occur in Staph aureus as far as we can tell. And again, you can't look at the whole diversity. Ideally, you would want to download all the MEC-A genes that are available on the whole NCBI. That would be like an ideal project to be able to talk about the actual diversity of the gene, right? So you take all of it and then build a tree and see what's going on. But that's a little bit, that's a little bit too much, I think. It would take much longer than a few weeks of the project to do that. Download million sequences of one gene and then align them, look at them. But yeah, I think the numbers is up to you again. You know, if you know how to do it for a large number, so you don't have to do it manually. So you know a little bit of coding and you can, you know, speed it up. You can do a little bit more. If you just can do like, let's say 5 or 10, that's also fine. Yeah, the paper I showed just 1,180 sequences, whole genome sequences. So that's what I used for that project. So nothing was done manually, everything was coded, hard coded. I didn't even look at things. Unless I build a tree, then I get annotated. But manually looking at SNPs and stuff, not possible. So you have to come up with a method to do it without actually physically seeing it, because you can't even open those files. So the large alignment file is about 90 gig. Can't open it. The text file of 90 gig. So you can't open it, you can't look at it. So you have to kind of find a way, a programming way to describe it if you've done everything correctly, if all the names matches and so on. Okay, so with FASTA as well, it could be that if you download multiple MEC-A gene FASTA files, the name in the FASTA could overlap because it's going to have just MEC-A. So make sure you check the names, that they don't have any special characters, they're not weird stuff. You can rename it yourself. So you can just put the strain name and underscore MEC gene. Don't use spaces as well.
And can I share my paper from like Google Docs?
Sure.
Instead of like sending? Oh, all right, thank you.
Yeah, I haven't had a chance to look at it yet. It's just busy. My student today ended hair project. I had a 10-week student, so she today submitted her poster and did the final presentation. So I was quite busy with that this morning. Are you sharing?
Yeah, I just shared it via Google Docs, like the Google, because I thought I had to send it via like a Word document and I didn't have the Microsoft. So I kind of like used the
Okay, no,
Google.
don't worry. Okay. So you send it as a Google Doc as well, right? So I've got
Exactly.
that. Okay, excellent. Do you want to look at it now together, or do you want me to give you a feedback after the session and just? So as I said to Hayley, Avahan is done, unless you're going to add some more stuff, Avahan, by Friday. You think you're going to add some more stuff to your report? Or are you happy for how is looking for now?
No, I'm like adding the alignment and where like common SNPs. I found like to had like similar SNPs in like number 637, I guess. So I'd add that figure.
Excellent. Yeah, once you finish it up, you can send it to me as well. And I will look at Haley's over the weekend and at yours, Rana. So if you update it by Friday evening, send me updated version with all the
All right.
results.
Okay. All right.
And I send you the feedback or edits or whatever on Word document or Google Doc, wherever you guys prefer, by Monday. So then you have some time until the next session to work on that a little bit. Yeah, I was thinking, did you had a session? Did Ash talk about discussion with you yet? Did you had a, how does discussion supposed to look like and what's supposed to be included in it?
Not yet. Like the last session, we talked about how to write the third part of the results with the UniProt and all that, but not discussion.
So that's the last part, isn't it? The third part of the results. Okay, so the next step would be your discussion. So I think it would be nice to have a little chat about it. So you start thinking about it because you did your literature review, you wrote your introduction, so you know the literature a little bit now. You're writing up results, and you know, once you write up the results, I want you to think about what they mean and how can you discuss them later the next session. So it's just something to keep in mind once you're writing it and thinking about it. So first of all, clarity of the results, tables, figures, make it clear, make it reproducible. That's the most important thing. So if I would give your report to someone else, would they be able to do exactly what you did? Okay, that's important in science. They have to be so they have to be described not into a detail which is crazy, but into the detail enough someone else with background, the same background as you, some simple ability to run bioinformatics, you know, tools and stuff, is able to take it and do it again. Okay, able to find the same sequences. So Rana, if you're comparing, let's say 20 sequences from other staphylococci, I want someone else to be able to find those sequences either by excision number. Does that make sense? Excellent. So do you guys know what do you think discussions should look like? Let's maybe start discussing from your point of view. Haley, do you know what you're going to put in discussion? What do you think discussion should look like?
I think like connecting the results and then the introduction.
Yeah, so to the literature mainly. So whatever is published on that, whatever been said, what other results are there? Are your results supporting whatever that's been said in the literature? So you're probably going to have to read 1 or 2 papers additionally for discussion. You know, it depends what you find. So if you look at within species, then you need to look at a few papers which did a similar thing. You look between species, probably with the tool comparisons. I'm sure there's a paper comparing the tools as well. I'm sure you can find a paper which compares all the different databases. So you can try to find it, but this is not as exciting. So I'm not going to make you do that and, you know, try to see if your comparative work. But for the actual 2 and 3, you can, if you're looking within a species, maybe you can find a similar paper about the MAC-A diversity, okay? So you can just link it to it. And let's say your results say there's a diversity or there's no diversity. Let's say there's no SNPs. Okay, you didn't find any SNPs. Then you can find a paper showing about, talking about how MAC-A gene is conserved and the SNPs don't arise. There's a strong selective, positive selection on that gene. It has to be the way it is so the mutation don't appear so much there. Okay, so it's kept the way it is to make it functional. Or if you find some SNPs, what those SNPs mean, what regions they in, what's the difference between different species. Maybe you would want to, if you talk about between species, maybe in your introduction you want to say which species actually have MAC-A, right? And then in the discussion you can be like, yes, indeed we find that those species which this paper mentioned that it has MAC-A, I also identify MAC-A. Does that make sense? Okay, is it just a practice? Normally you have like a groundbreaking result, you know, it doesn't happen then often, but normally you have like very exciting result and you need to frame it, you need to sell it, you need to make it significant in the context of what everything else is there. And you also have to show wherever you did, it's different to what other people did. Because if you did the same thing other people did in the literature, there's no point publishing it again, right? So you need to show how your research fits with the literature, but also show it's unique, it's interesting, you've got interesting results. They deserve to be published, they make sense, they're significant, they show interesting patterns. Okay, so yeah, I can, you know, I don't have a discussion. Is there a discussion in the review? I can't remember. But you can have a look at some example papers which do that. And again, it's hard because you need to know the whole paper in order, you know, what they did exactly in order to think about discussion, right? But that's where your project is. You have to think. So examples might be hard unless you want to read the whole paper, the whole results section, introduction, and try to, you know, think of what you would talk about and then see what they talk about in the discussion. Just see if you could, you know, even have an idea what's going on in that discussion. We could do that. Cool. Okay, do you have any questions? You can reference new references in your discussion. You don't have to only use your introduction references. So you can add papers. Don't have to, but you can. Okay. I would do a quick literature search for the discussion. You know, once you have your results, maybe read 2 or 3 papers. I don't expect more than that, just to put in a context. But I would think your introductory references might not always fit to what you find. Okay. Okay, do you guys have any questions this stage? Any questions about table, figures, referencing, title? Do you guys have a title yet for your report?
I was
No.
gonna ask, like, should we like target specific mutations and like kind of see what it does to the structure of the protein, or like can we also talk about how hypothetical proteins, like there are some hypothetical proteins that can cause methicillin resistance, and like they are found in these species, and that's why we should, like, I guess, raise awareness and etc.
Okay, so the protein annotation is a separate thing. There are loads of tools which annotate proteins. They don't always work. So they have a strict cutoff. So occasionally it doesn't identify MECA gene as a MECA gene, or it doesn't identify, let's say, penicillin binding protein gene or other genes because they're too divergent. So they're too different. And the annotation pipeline doesn't account for that. So even if something is called hypothetical protein, it doesn't mean it's hypothetical protein, okay? Always. If it has a proper name and is properly justified in your product, it means it has been tested in a lab. But it doesn't mean that someone didn't test if that strain is methicillin resistant or sensitive in the literature, if it's potentially a hypothetical protein. So I wouldn't just think of a gene and how similar it is, and you can talk about it was annotated as a hypothetical protein, but then you can look into the literature if that strain was tested for MIC. Okay, so we talked about MIC. Okay, so that's the cutoff for MIC. So if you're interested to look at the phenotype of that strain, there's a possibility there might be data there available. It might not be. But, you know, overall to get an idea, so then you can, you know, there's also like sometimes when you look for strains and then CBI gives you like a table of MIC data for that strain. I don't know how to find them. They sometimes come up, so I never looked for it specifically, but sometimes I see that. So sometimes you get a strain which has the MIC data, you can think about it. And there's, you know, you would have to look at the UCAS website to know which cutoff is resistant or sensitive, right? We had that in the lecture. So there are cutoffs which above certain value is considered resistance, below certain value is considered sensitive to the drug. It's to do with the minimum inhibitory concentration. So you could look into that. It's a lot of stuff to look at. So Rana, I would write it up, the main bits, try to do a little bit extra, see how you get on, because the time is running low. It's a lot of work. So try to get the basics done first. And I'm happy for you to build on top of it, no problem. But it's good to get the basics first so I can read through it. So maybe if you could go to put the basic section by Monday-ish or by Friday, Saturday, so I can read through it over the weekend, that would be great. Sounds good? Great. Do you guys have any questions? We've got 20 minutes. We can talk about the report, or we can talk about any questions you have from the last lectures. Or I can show you some examples of things if you want, if you're not sure. Haley, anything?
I think I'm good right now. Thanks. So.
Okay, great. Avahan,
No, I don't have for now.
you happy for now? Okay. Rana? Oh, good. Okay, we've got 20 minutes. I need to think of something to do if you don't have any questions. I've got your report, so I'm going to look through them, but I do it in my free time. We're not going to do it together. So we could look at more kind of discussion concepts, if you like. So we could look at how discussion looks like.
Join another Zoom at 11:45. Is it okay if I leave a little bit early?
For the next session, so the Saturday session or today?
Right now.
Yeah, that's fine. If you have to go, that's fine.
Okay, I'm gonna miss
No
anything important.
Yeah, just remember to send me the Google link. Let me just check if I got it, maybe. I haven't
Okay, I
checked
shared it with your email.
Yeah, great. Let me just check if I've got it, and then, yeah, I've got it. Let me just click on it if I can open it. And if I've got the privileges. Okay, I've got the editing. Okay, excellent. Thank you so much. So I'm going to use the suggestion mode, so then you can choose to either accept or reject my comments or changes. Sounds good?
Thank you so much.
Excellent. Okay, I will see you. I think it's Saturday next time we see each other. Excellent.
Okay, have a nice day.
You too. Take care. Okay, do you guys want to look at some discussions, examples, and have a little think how they look like?
All right.
Yeah, okay, I can show you my current paper, which I'm working on. So I showed you some figures. This is actually about MEC gene. It's kind of relevant. It's not directly. So what I did in that paper, so just to give you like a brief thing. So I looked at there's a clonal complex called CC398. Did you came across it when you did some literature reading about MEC and SC-MEC and no?
I didn't.
No, okay. So CC398 is a clonal complex. So the population Staphylococcus is separated into different clonal complexes, which are like clonal population expansions kind of things. This specific clonal complex historically switched between human host. It jumped to livestock species, mainly pig, and it was there for kind of 50, 60 years. So kind of switched the host, adapted to the livestock host, picked up loads of resistance, including methicillin, penicillin, and tetracycline. Penicillin actually was present in human, mainly methicillin, tetracycline. So picked those two resistance genes on mobile genetic elements, and then it became successful. And now it's able to jump back to humans from livestock. Okay, so instead of having strains which are sensitive infecting humans, now we've got these highly resistant strains. But the resistance appeared in the livestock population. Does that make sense?
It does.
Okay, cool. So what I did in that paper is I tried to reconstruct the evolutionary history of the element those genes were carried on. And what do you think that would be useful? Do you have any idea? So you know SC-MEC, so you read the paper, so there's a different mobile genetic elements, right? The MEC sits on it. So why would I use the whole element rather than the single gene?
Maybe to track down its evolution and like predict what it could become in the future.
Mm-hmm. Yeah, predicting, yeah, if it's still stably maintained in population for a long time, you would expect it will stay that way. Yeah, that's good. But why would I not use single MEC gene as you guys do? Why would I use the whole element?
Maybe kind of like researching about the relations to the other genes.
Yes, sure, but why not single gene? Why would you think not single gene wouldn't tell anything?
Because like when you look at a single gene, you basically just look at its function, but not its relations or like its evolutionary relations, I guess, with the other genes, because like some other genes could.
Yeah, but you have a gene. Okay, so you've got a bacteria. MEC gene entered the population, right? It was present in the livestock clade. So why can I not reconstruct the phylogenetic history of that gene? Why do you think So I did say to you, if 50 years ago, roughly, that clade appeared. So 50 years is
Maybe
a
it
long
could be
time, evolutionary
different.
speaking. What do you think? For single gene evolution, is 50 years a long time?
Maybe I don't think so.
No, it's not very long time for single gene. So if you have a gene and you need the gene and it does important things, then there is some pressure on it to keep it that way, right? So if it mutates in a bad way, you lose the gene, right? So if you want to keep it, so you keep the gene, it's important for your survival. It's less likely to get mutation in the gene as well, so that the evolutionary pressure on that gene is high. And also 50 years is not very long time, okay? So for like just, if you're part of core genome, there's no recombination going on, just accumulation of SNPs in a short gene, because MEC gene is quite short gene. It's like 2000 base pairs. Avahan, about that, right?
1007, I guess.
Yeah. So it's a short gene. So you wouldn't expect, when you calculate the rate of mutation, looking at Staphylococcus aureus, you can do that, right? So if you have a phylogenetic tree of Staphylococcus aureus, you've got an idea how many mutations per year do you expect in that population. You know, when you calculate it on the whole genome level, you're very unlikely to see, maybe you see 1 or 2 SNPs in that gene, okay? Over 50 years. So then 1 or 2 SNPs is not very good, right? Because 1 SNP doesn't tell you much. 2 SNP, you know, when you get this 1 or 2 SNPs and they randomly distributed, they're not in like a phylogenetic, you don't get 1 SNP and then after 20 years you get other SNP, but you don't get any other SNPs, and it's clear, maybe 2 SNPs enough to say something, but it's not very much, right? Do you know what I mean?
Yeah.
Yeah, so in our context, you know, you're looking at single SNPs, you're looking at the functional side. But if you'd want to, let's say, ask a question, was this gene maintained within that population? You'd have to reconstruct the phylogeny of the whole organism, right? Using like a tree building, alignment, all the stuff. But you also would want to do that for that single gene. But that single gene would have to have enough change for you to be able to infer its evolutionary history. If it didn't change much, there's not much you can say, right? Does that make sense, Avahan?
Yeah.
Okay, so now what? Now you know that. So why would you use the whole element? For that reason, right? So you have the whole element, you've got more genes, you've got larger fragment of the genome. Also within that element, you would have genes which are not so important, not all like, you know, super core. You're going to have intragenic regions, you're going to have the recombinases, you're going to have quite a large part of DNA, right? So you can just reconstruct the phylogeny using that element, right? If everything works well. So if the SCMAC, even so, you know, 30, 40 years, there's loads of rearrangements because it's a mobile genetic element. You know, it's transmitted horizontally. It's prone to rearrangements. That's what we see. Okay, with the element which was transmitting tetracycline, TN916, we don't see any changes in the structure of it. We see SNPs, but we see enough SNPs to be able to say those SNPs were acquired within that population. Does that make sense?
Yeah.
Excellent. Okay, so how would you, okay, so you know the results. I told you there's 2 genes which got maintained. What would you try to talk about in the discussion here? What would be the obvious things to talk about in discussion? Okay, so we've got gene maintained in a population for 50 years and the second one for 30 years. Very long time. You know, they very rarely leave the population. They stuck in there. They're present in livestock clade. Even so, livestock was treated with drugs, loads of antibiotics historically. Now, not so much. The usage has decreased, right? We're more aware of the antibiotics leading to crisis and antimicrobial resistance. The livestock is not treated with so many drugs anymore in most countries, right? So what would be the discussion here?
Maybe I can't really think of like a discussion without knowing the exact results, but maybe like connecting the results to the general usage of like livestock and where, I don't know, resistance could occur, like because there's like specific countries. in, like,
Yeah,
livestock.
yeah, no, that's a good point. Linking it to usage per country, seeing which country the CC is most prevalent, what's happening, that's really good. And also trying to think about how does our result have an effect on future, right, result. So let's say we showed there was a known decrease in antibiotics, however, the element was still maintained in the population. So even so the drug was not used against this population anymore or was less frequently used, the gene got maintained within the population, which means simply decreasing antibiotic usage on farms might not necessarily mean you're going to get sensitive populations. Does that make sense?
It does.
Cool. So it's just like an idea. So I'm just giving you a very short example. So this is paper I'm working on. It's much more to it, as you said, much more results, much more data. But it's just trying to think what kind of implication our results have. And that's the main one for us. We show maintenance. No one ever showed that maintenance. The first one showed such a long time maintenance, and that's the main result. Okay. So you're trying to put that in the context of literature and what does it mean as well for treatment, for prevention, and it's limiting, right? If the maintenance is occurring in a population, that's really worrying because actually decreasing the drug usage won't have an immediate effect at all. It's going to take a long time, or I don't know how long because we haven't seen that yet, but 50 years or more, okay, to lose the element at least. Cool. Any questions about that?
No, I
So what
guess
do you,
I'm.
yeah, go ahead, sorry.
Oh, I said I'm set.
Okay, so what do
Thank
you think you're going to put in your discussion then by looking at your results? Or what kind of papers you're going to have to look at to try to understand, to put your results in a context? Avahan?
Yeah, maybe if we I don't know about the first analysis, but about like other species that we found, then we could like really look into what are the different kind of risks that like these bacterial species other than SRES like have to our health, and how could they arise as a new superbug, like how much potential do they have, so looking onto that.
No, that's really important. Yeah, if you find mecha in a species you don't expect it to find, yeah, trying to understand how it got there, great. Yeah, looking at the literature, what was said about it. And again, it's really hard because you need to read a lot in order to understand it. And, you know, as much as introduction results are important here, I think I want you to think about your discussion once you write your results because it's super important. Because discussion is what sells the paper. Discussion is what puts your research in the context of literature, is the most important part. Okay? And we don't expect you to read 20 papers and try to put it in context of them. You know, it's with like research paper, obviously you have to read it all there is on that subject. So if you miss like a paper about it because you haven't found it or you haven't, you know, you ignored reading it, then you might be in trouble because let's say the people who's going to review your paper, the papers wrote that original paper and you're just kind of not including. So you have to have comprehensive literature review of the subject every time you write about it. If it's something detailed, obviously you can't know everything about Staphylococcus. There's 25 papers coming out every day on NCBI with Staphylococcus aureus in the title. So reading them all is literally impossible, right? But you can read all the papers on CC398. So that's the clonal complex I work with. There's 320 papers only. So that's a good start. And again, you can skim through the abstracts and see which ones are more relevant to what you did. So that's a good starting point. Do you know how to look at papers to see if they're good? Have you managed to, like, if you find a paper, how do you justify, you know, how do you think if it's a good or bad paper? Because there are bad papers, there are good papers, not all papers. Even published papers are not all great. They're all good and decent for publication for certain journals, but there will be a whole range of different papers, right? And some people do things
Generally, look at
differently.
the abstract. of the paper.
Abstract. Yeah, it's a good start reading an abstract, but there's also something you can look at on Google Scholar on CBI is the citation record of a paper. Okay, how many people actually cited it in their research? So if the citation record is really high, that would suggest it's a well cited, well published, well read paper, right? So for your references, you can have a look as an exercise. You know, if you have a few minutes, you know, copy the title into NCBI or Google Scholar and look how many how cited the paper was. Okay, and there's no like limit, if that makes sense. It can be cited as many times as it wants to be. But if it's cited 3 times and it was published 10 years ago, that's a bit, you know, not many people, 3 people only read it for 10 years. If it's published last year and it has already 50 citations, then does that make sense?
It does.
Yeah, so look at the publication date and then the citation record. Because if it got published last week and it has 2 citations, that's already pretty good, right? But if it was published 20 years ago, then not so great. Cool. Any other question? Any last bits you want to ask for? Are you happy to crack on?
No, I think I'm okay.
Okay, good. Okay, try to get as much as done as you
don't
can.
have for now.
Excellent. Thank you, Avahan. Try to get as much as you can done by the next session. I think because we're going to have one last session and the one before last would be then that's the one I can give you some feedback on what you already have done. So it would be good to have that kind of results by the next session all done. So you can have a little think about discussion, look at your results as well. So try to make the plots and tables, and then we can have a look and compare at the next session what are results showing, because you're probably going to look at different aspects of the data. It would be interesting to see what you come up with and try to think about it and look at the literature. I also look at the literature, which papers are relevant. I know the literature a little bit on MEC-A and SCMEC, so I can always help you out with if you're thinking about your results and try to put them in a context, what paper would be the right paper to use in your discussion. Sounds good?
It does. Thank you.
Great. I'm waiting for your reports. And yeah, I will see you next session. And good luck. If you have any questions, just message me on Slack. Okay?
All right. Thanks.
Cool.
Bye.
See you next time. Bye.